It seems big to propose that we get out of the problem of causation. Typically, we're satisfied with looking at correlation. And so intuitively, we think that when it comes to a cause, what we need to do is to intervene. So we have to do another experiment. We can't just look. And I will argue today that is not always the case. And you can actually get a stronger version of causality, run the fancy technique that I just talked about. Each time the input neurons are active at the same time, we get the indication that there's a ternary interaction. I really appreciate that you showed up, even if it's virtual. So first of all, I want to thank my laboratory. I also want to thank now Tsuchiya, who 2 or 3 years ago really changed my life by setting up a YouTube channel and let me just witness his lab and inspired me to go into the directions that I'm going. I want to thank Giulio Tononi and people in his lab. There's more that I could mention here that have helped me in understanding some of the things that I'm talking about. And I want to thank James Watson from Oregon State University that we're collaborating on some of this work, as well as 2 mathematicians, Carlos Zapata Carretala from the Wolfram Research Institute, is also the founder and president of SEMF, and Johannes Kleiner from Bamberg University. This having said, if there's any problems with the mathematics that I'm presenting today, that is all my mistake. And I want to thank Thilo and Andre and other people in the department with whom I shared discussions about what I'm going to present today. And last but not least, the work that I'm doing currently heavily rests on open science and open software tools. So I just want to mention that these are free tools that I'm using, that everybody is welcome to use. And all of the code that I'm talking about is available on Colab. Everything is free and open. So if you look at this logo, this was the official logo of the 2014 World Cup, Soccer World Cup in Brazil. And if you look at this World Cup logo, you might recognize that this is what you can win if you win the World Cup, this certain trophy at the end that roughly looks like that. And so this is a good example in vision of what we would call a purely straight feed-forward process, in that we think, well, it's not purely feed-forward. Of course, in interpreting that, your brain will also make use heavily of feedback. But to show you what feedback can really do to a feed-forward process of seeing things, I want to show you very quickly a facepalm emoji. You might now recognize that this almost looks like there's a facepalm emoji that was baked into the cake right off the get-go of this Brazilian World Cup. And so that is an example of feedback, of course, because I had to show you the facepalm emoji, tell you what the facepalm emoji is. And so other parts of the brain had to then provide this information to the visual system and modify it. And that is relevant to a lot of the research that me and others in the department are interested in, because we believe that now that we have the ability to look at neurons across the different layers of the different compartments of cortical columns, we can maybe separate out what is feedback and what is feed forward. So in particular, this right here is a model that Andre Bastos published in 2012, showing you from the top of cortex to the bottom, so layers 1, 2, 3, 4, 5, and 6. That is the standard circuit, the standard model, as we call it, of how neurons in cortex are put underneath and up. The important part is that feed-forward part, the information that enters the brain initially from the eyes, is vastly limited to layer 4, right in the middle. It's the meat of the sandwich, if you will, whereas feedback is mostly hitting the layers above and below. And so that poses an interesting question. So if we have research questions and we're asking, well, is feedback involved, or is this feed forward only? We can ask, for example, well, what affects different neurons? So if we have neurons in layer 4, 5, and 6, let's say 5, 6 neurons, and we're asking, well, what affects them? What is driving them? We can ask, is it layer 4C, where there's really the bulk of the feed forward information, or is it coming from layers 1 to 3, or layer 1 to 4B to be exact, where the feedback is coming from? Now, that's a question that a lot of people are interested in and like to ask, but there's a more complicated scenario here, which is that it could also be both. You could have feed-forward and feedback information activating neurons in layer 5, 6 at the same time. So if you don't have a clear-cut contrast where you can say, look, this is all middle layers, or this is all coming from upper layers, this gets much more difficult. So what I'm saying is that we might more often than not encounter scenarios where the simple scenario of having a clear-cut distinction falls apart, and what we're actually looking at is a combination. And of course, that's still interesting to look at. So, but if we're asking, so how much do feed forward and feedback cause responses of, say, neurons in layers 5 and 6? that is what we're interested in, then we're facing two dilemmas. The first one is when we're saying cause, well, then we can't stick to correlation. So that already rules out statistical tests. And when we say, is it both the feed forward and feedback, or both the activity of layers 4 and layers 1 to 3, then a technique like Ranger causality, which tries to get us out of pure correlations or associativity and gives us an idea of quality, it doesn't work because that technique is pairwise strictly limited and it can only speak, is it layer 4 that's causing the activity in layer 5 and 6, or is it layer 1 to 3 causing in layer 5 and 6? It cannot deal with the scenario if there is a mix of. So I want to talk about both of these problems today and propose a potential solution that I'm quite excited about. So it seems big to propose that we get in science at all out of the problem of causation. So typically we're satisfied with looking at correlation. And so intuitively, we think that when it comes to a cause, what we need to do is to intervene. So we have to do another experiment. We can't just look. We can't just look at data and collect data. We have to inject maybe a pharmacological agent or use optogenetics or something. And I will argue today that is not always the case. And you can actually get a stronger version of causality without doing that. So that is a very, I know, controversial take, but it's actually in the field of causality quite established. And that is usually done by looking at counterfactual. So I will come back later to this point, but basically there's a stronger version of causality than even interventions allows us to do. And that is if we know everything that could have happened didn't. So that's one of the things I will come back to that. So if we do that and we established causality, let's say in a neural system, then we still have to quantify it or measure it. So I do not just want to show you that there are certain causes, but I also want to be able to quantify them, measure them. And then, of course, lastly, the question is, how do we deal with the fact if we have a combination of causes? So if there's several things causing at the same time. And so, in fact, it's this combination of causes that I will mostly spend my time on, because I think that might be an underappreciated point of brain function in general. So what do I mean by that? So most of us, that's how we think the brain is actually working on the most fundamental level. So you have neurons and they are connecting. There's a little bit of a gap between them usually, and they each are electrically active. They release chemicals, and that's how they activate other neurons. So let's say neuron A is innervating neuron C. So neuron A is an upstream neuron and neuron C is a downstream neuron. Well, of course, in the brain, this is never that simple. So we're not having A-B neuron pairs. We have, or AC neuron pairs. We have neuron A innervating neuron C and neuron B innervating C. So of course, it's this tangled mesh that we're dealing with. And so one of the first people who really thought about that quite deeply was Donald Hebb. And in fact, he did that as a PhD student. So I found this quite interesting. He published this in his PhD studies. And what you can see right here is a drawing that shows pretty much what I just showed, a slightly different label. But you see how neuron A is innervating neuron C, and then you have neuron D instead of neuron B innervating neuron C. And so most of you are probably familiar what Donald Hebb was getting at here, because that is still what we call a hebbian synapse and hebbian learning. This is underlying a lot of what we still think learning happens on the neural level. And it is, of course, the basis of a lot of what AI and machine learning does today. So in fact, what I'm showing you right here is a schematic or cartoon that I took from the machine learning literature. And it shows pretty much what I showed before. So we have two presynaptic neurons, neuron A and neuron B, and a postsynaptic neuron, neuron C. So I will use presynaptic and upstream, or postsynaptic and downstream interchangeably. And so then what Donald Hebb postulated, and so what is used in machine learning, and again, what most learning theories rely on, is that you can think of having a causal effect that can be quantified, synaptic strength, and here as the W. So let's say neuron A has W31, so that's a certain weight, if you will, of the synapse. That's how much strength or power, causal power neuron A has. So you can think of that probabilistically. If neuron A fires, there's a certain likelihood that neuron C might also get active later on. And so if that synaptic weight or strength is higher, then the likelihood of neuron C firing after neuron A fires goes up. And of course, neuron B will also have a certain likelihood or synaptic strength or weight onto neuron C. So we have this very simple scenario where neuron A and neuron B is active, and then with a certain likelihood, neuron C becomes active and produces its own outputs. Now, of course, what Donald Hebb, though, said is there's an interesting scenario here, which is what happens if neuron A and neuron B are simultaneously active? And that is, of course, what we now usually think of in systems neuroscience as synchrony. It's also, embedded in our ideas about coherence and other ways about population activity. And so that is an interesting case because it could be that we know exactly what the synaptic weights are, the strengths are. Let's say neuron A on neuron C and neuron onto neuron C. When both of them are active, something else happens. So if both of them are active, the likelihood of neuron C firing might be higher or smaller than any of the numbers or the combinations thereof that we found earlier when we just looked at what neuron A and neuron B do. And the implications of that are actually quite profound. Now, Donald Hebb used this as an example of learning. He said if neuron A and neuron B are active simultaneously, then maybe that changes the synaptic weights, or it has some other effect on neuron C, and hence the synapse might get strengthened and learning occurs. And so that's why when you see red and you hear apple, these two things often go hand in hand, and that synapse gets more and more strengthened. But I will make a different point today. What I'm arguing is that if you have the case, if neuron A and neuron B are active at the same time and the effect is different than what each of them do in the isolation or any combination thereof, that means we cannot study the system, or I will say it more strongly now, we cannot understand what the system really does by just looking at each of these neurons in isolations. And I will say that this is a problem. Now, the first question that I usually get when I introduce them to people is they say, well, it's true. So could it just be that we can even infer combined effects? So yeah, maybe it's not just that they sum. Maybe it is some other combination, multiplication or normalization. The first thing I want to point out here again, these are theoretical models of a Hebb synapse, and you can see that the standard assumption is usually that all of the inputs just sum. So if, let's say, you have a certain probability for C to fire after A fires and a certain probability for C to fire after B fires, you just sum their probabilities, or maybe you multiply their probabilities. Now, I want to show you some actual data. And to show you the actual data, I just will use one of the most simple systems. So this, of course, here is a very famous cartoon from David Hubel and Thorsten Wiesel. And that shows that in the LGN, we have, that's the first output after the I, we have neurons, and they are thought to converge onto V1 neurons. That was their assumption because they found that the receptive field structure makes sense if that's the case. And so I would just point out here that is very much what I showed in the beginning. So we have two neurons, A and B, and they fire on neuron C. The only difference is that neuron A and B would be in the LGN and neuron C would be in the primary visual cortex. But again, if I use this Hebb diagram, same thing. We now have two neurons, A and B, they're in the LGN, and neuron C in the primary visual cortex. Now, this would be a great study to do, let's say, in a primate. But it's very difficult. It's already difficult to find neurons in the LGN, to hold them for a long time, to also have an electrode in V1 at the same time. So speaking from experience, these are heroic experiments. But people that succeed and then also are lucky enough that they find neurons that have overlapping receptive fields, they get rewarded. So this right here is a Nature paper where that actually happened. And it's a great Nature paper by Jose Alonso and Madi Asri when they were in Clay Reed's lab. And so I thank Andre for making me aware of this paper. And what they show right here is a little bit complicated, but it's basically what I tried to set out. So A and B and C, what you see right here, are receptive fields. And you can see the receptive fields in the LGN, they're concentric, as you would expect. And the receptive field in V1 is roughly linear. And they knew that these neurons actually connected because they computed cross correlations. So whenever the neuron A fired, they would look at when does neuron C fire? And they found that there is a peak in a correlation, and it happens exactly 8 milliseconds later. Now, why is that important? Well, it takes time, as I said before, for the signals to travel from the LGN to V1, and then it takes time for the chemical transmission to happen, and that is typically in that time range. And that's important to note because it means that nothing else can intervene. So what you see right here is the effect of neuron A onto neuron C. And then they really got lucky to find another neuron where that's also the case. So they had this golden scenario where they had one neuron in the LGN and another neuron in the LGN, different neurons, and they both were innervating the same V1 neuron. Well, now they could ask this question that granted them the Nature paper. So what is the causal efficacy of these neurons? And they did it very simply. So they just said, what is the percentage of thalamic spikes, let's say for A, followed by a cortical spike of C? And then they compared it to a shuffle correlation. And so what they found is that for neuron 1, they found the likelihood was 1.71%, and for neuron B, it was 1.41%. But the kicker was when both A and B were simultaneously active, this efficacy jumped up to 5.28. So the sum is 3.12, as you can see. So that means that they found this really striking effect that indeed, if you have neurons that are arranged in this way in the brain, they can form a effect that is not inferable if you just study the individual pairs that the system is made out of. Now, typically, when I present these data, the next thing that I hear is, well, maybe they got really lucky, and there's only one of these synapses in the brain where this really happens. And what we can usually do is another diagram to show that is we can totally just look at what one neuron does to another, what another neuron does to the same neuron, and infer. And this thing that I'm showing right here, where both of them are active at the same time and you get a different effect that is very difficult now to show diagrammatically with lines, so I'm just using a surface here, that maybe doesn't happen that often. Well, it turns out that this, what I again symbolize here with a triangle, because it's basically a ternary effect between three neurons that is not sociable into pairwise effects, into lines, that is actually the most common case. So when I said in the beginning that they got very lucky, I actually didn't quite tell you the truth. What actually happened is they worked really hard. So they did so many recordings that they found in the end 13 of such neuronal triangles where they had two neurons in the LGN and one neuron in V1. And out of these 13, 11 of these triangles, they showed this effect. Then when both of the upstream neurons were active, the downstream neuron was more likely active than could be inferred by multiplying, summing, or any kind of straightforward combination of the individual effects. So again, here's just a simple diagram to show what I mean. So if we translate this into the language of graph theory that a lot of us are familiar with, and now the neurons are just circles and the axons, if you will, are just arrows. You can think of these arrows as weighted edges and the neurons as nodes. Then we get a certain likelihood, a transition probability from neuron A to neuron C, another one from neuron B to C. And so I'm using capital letters to indicate, and also the color yellow when they are active. That we can easily do with graph theory, but when both neuron A and B are active at the same time, this language falls apart because we've used all of our edges. We've already used all of our arrows to show what we mean. So what I'm doing, I'm making another edge right here. So this edge that spans all three of these neurons, that's showing that when both of them are active, something else happens. It's a ternary effect. So what I'm going to argue for the rest of this lecture is that quantification of binary relations is insufficient if the system has such synergistic effects that are not easily coming about by looking at the simple components. And so I will argue that that is a little bit of an overlooked problem, and I will argue that that lies in the fact that mathematics itself is very streetwise. So just a typical argument right here. Here you see typical data from a lot of our publications, and you will see, if you look closely, that this is fundamentally always looking at pairs, looking at different conditions, for example. And you compare t-tests or ANOVAs. Correlations, of course, are fundamentally two-dimensional. Range of causality are already mentioned. And I think that our thinking about the brain that has shifted in the last 10 years or so, 10, 20 years, towards thinking of the brain as a more connected network, when we use that language, but then think about it, we actually usually tend to think of it as a graph, as shown right here. This is often what is shown in literature. And note that these graphs, again, are strictly pairwise. All that you find between these neurons are lines. They always connect two neurons. Of course, each neuron can be connected to many other neurons. I'm not saying that there's only pairwise connections, but what I'm saying is it doesn't account for the fact that if two neurons are active at the same time, that things change. And with simple lines, we cannot even symbolize this here. And then lastly, and this came as a surprise to myself as I sunk myself into this topic, most of mathematics has the same problem. So fundamental arithmetic, if we think about subtraction, multiplication, and all of these, they are fundamentally binary. There's always two numbers that get operated in this way. So when we think of natural numbers, for example, in addition and multiplication, what we actually do is we always just deal pairwise. And so this can maybe be a little bit unintuitive, because if I tell you what's 2 plus 2 plus 2, you might think, oh, I can just take these three numbers and immediately add them together. But I'm going to argue that that's not really what you're doing. You're just actually memorizing what the result is, and you're taking a shortcut. Because if I give you 3 larger numbers, then probably this will fall apart. So if I tell you 16 plus 16 plus 16, what you will probably do is you will first add 16 to 16, and then you will add another 16 to that. And so likewise, if I tell you what 7 times 7 times 7, or 7 cubed, you will probably first compute the square, and then you will multiply that with another 7. And it's interesting that we do that, because when I looked more deeply, I found that that is true for every single mechanical calculator that has ever been invented, ever since the early days of Leibniz and other mathematicians. And in fact, this is also true for every single von Neumann computer, like the ones that we all sit on. So the logic, the Boolean logic that all of our computers rest on, you might think that if you add 3 binary numbers together, maybe there's a way that you can combine 3 binary numbers at once. Maybe a GPU does that, or one of these new machine learning. No, because everything boils down to AND gates and OR gates, and in fact to even more simple types of logic. Fundamentally, we're dealing with pairwise operations, and it goes even further. So just to summarize this point that I was making here, it seems that it should be easy in mathematics, that we have to the left-hand side shown here many different things, n different things, and we have just one operation where we combine them all into one. But what we actually do is whenever we do that, in particular, as I just said, with natural numbers and addition and multiplication, what we actually do, we always take the combining many things and break it into pairs, and we do it one pair and another pair and another pair. And that's how we can actually do it, that we combine many more things. And without getting too much into the mathematics here, the reason for that is that numbers, all the numbers, the real numbers that we usually deal with, in fact, all complex numbers, and so that, as you know, entails all the rationals and other numbers we usually deal with. Mathematically, they're all fields, and all fields are rings, and all rings are groups. And so that means they share the same axioms. And so the fundamental axiom of a group is that it has a non-empty set and an operation that combines any 2 elements. So it's by definition a binary operation. So Yoda was right, always 2 there are, not more, not less. And so this will be a little bit of a tangent, but I have a hunch, or I'm going to maybe get out of on a limb here, why this might be the case. That so much of our thinking, in fact, as you can see, so much of our core mathematics are fundamentally pairwise or binary. And that's very recent data from the hippocampus. So this is research that I wasn't fully aware of, that is actually really striking, that has arisen out of people trying to finding that has in the rodent field been established for many decades now and yielded a Nobel Prize, which is that there are place cells. That if you look in the hippocampus, and you let a rodent run around in a certain maze, then you can find these neurons. They form basically a hexagonal grid, and individual neurons are active depending on where the neuron is. And so Carey has done very interesting work on that, for example. And if a mouse dreams, maybe it's running through the same maze again. And indeed, it seems like you can read a mouse's dream out of the hippocampus using these grid cells and these neurons. Now, for the longest time, I was aware of that. People struggle to find that in primates and humans and so on. But there's been a student breakthrough where people found, and this is what I'm showing right here, that not only do you find these kind of grid-like organization in the mammalian, in fact, in the human hippocampus, but what you find there, it goes even further than that. So what you find right there is that you find these grids not just for location, but for cognitive spaces more general. So if you have 2 axes, let's say right here you have vehicles and then size, you can find that vehicles are organized in the hippocampus by size and vehicle type. So without getting too fancy, what we're dealing with here is a Cartesian product. So if you have 2 sets and you look at all the combinations of the sets, that is a Cartesian product. That is why we have a coordinate system. That was René Descartes' discovery. And it seems that what our brain does here with this neural machinery in the hippocampus is fundamentally when we do these things, is to start literally in the hardware sorting things out pairwise by having these 2 dimensions. That is the data where it stands. And I know that people are looking, can we extend this to 3D? And looking in bats where the space that they navigate in should maybe be less 2-dimensional and flat as it is for rodents, and should have more of a 3D component. And what I hear is that's very difficult, and maybe results are even a little bit unclear. So it seems that at least when it comes to us, maybe this pairwiseness is baked into the cake when it comes to our thinking. And you might wonder at this point, well, maybe there's no problem with that. So if evolution bestowed us with this binary thinking, maybe that's because each time there are higher order interactions, we can break them down into binary interactions. And that is a discussion that is legitimately to be had, and it's a discussion that is quite active in the field of mathematics, and there's actually some indication that that might be true, at least in certain general, very theoretical, abstract mathematical realms. I'm going to argue that when it comes to physics and science and the world, though, this is not the case. And so here's a very simple example, which is the combustion motor. And so you're all familiar with that. In order for your motor to work, what you need is you need to have air and fuel, spark, all come together at the same time. If you have just the fuel and the spark come together, nothing happens. If you, again, have the air come too soon and you have just the spark and the air, but no fuel, any of these combinations by themselves are ineffective. What really has to happen is it has to happen exactly at the same time, and then something entirely new happens. This is one of the few examples that I find plausible of the effect of emergence. But also note again that it means you have something where three things come together and something new happens that none of the pairs can evoke, that it cannot be broken down into pairs. When you analyze it in pairs, you will never see what happens once you look at what the three things do together. Okay, so what I'm going to argue that maybe when it comes to our neural data analysis, what we want is not binary mathematics, but we want to be able to do ternary or quaternary or n-ary mathematics. And so going further right here, I said it's mathematical too as a binary, but there are many exceptions. So there are n-ary groups, there are tensors, of course, you might be familiar with. So in linear algebra, some of these problems have long been solved, if you will. So there are n-ary algebras. There's also hyperstructures and hyperoperations. And then there's something that I will talk about for the rest of this talk, which are hypergraphs, because we already talked about graphs and it's a simple modification. All right. So, okay, so just real quick, of course, the actual problem with dealing with the data, there's some more caveats that maybe, again, I will talk about in the end. So let me jump over this quickly and make it more simple. Go to the simple example of the Hebb model. And the first assumption that I'm going to go over a little bit quickly maybe is another objection that I get when I present this with people say, well, this is a nice theoretical example, but it never happens in the brain, because all of these neurons probably get inputs from other neurons. So to isolate three neurons like that is already unfair. And so I argue that for our operations, that's actually, we can do this at least to some degree. And so I'm going to justify this real quick, a little bit abstractly. So in the field of thinking about graphs causally, which I think some of the greatest breakthroughs have been done by Julia Pearl, and you will hear his name a couple more times, this idea of a Markov blanket arose. And so you can see right here a graph where you see by the arrows that you have causal interactions from the top to the bottom. And so in the language of causal graph theory, we would call the upstream neurons or the presynaptic neurons in this graph the parents. So this is where the causal effects emerge from. And the parents, they affect descendants. And so what a Markov assumption, or what this idea of a Markov blanket comes from, is from the Markov assumption that states that every node in such a network, if it's a Bayesian causal network, is conditionally independent of its non-descendants, given its parents. So what this means is that when we look at the causal effect onto a certain node, what is affecting it, the only thing you really have to worry about are its direct causal, are its parents. And so even though the parents have their own connections that you can basically ignore for the moment. So what this means is that if you have, and again, I'm going to use a little bit more complicated language here, if you have a directed acyclic graph, which just means that there's no loops in this, and that's a Bayesian graph, it's a Bayesian network, a Bayesian causal network, what you find is that each node is conditionally independent of the entire network in the sense that it only depends on its Markov blanket, which are its parents. So where the effect is coming from. So the causal Markov condition means a node is independent of all variables which are not effects or direct causes of that node. So that I'm going to use a little bit, roughly speaking, justifies that we can isolate certain input-output relationships of neurons with some justification. Of course, it's not perfect because we don't have all the parents, we don't have all the descendants. I'm also immediately going to say that, yes, that is not perfect. So it is a caveat that remains, but all I wanted to say, it's justified to some degree. So I'm a little bit fuzzy, and that may be okay, that we can look at these isolated neurons. Okay, so at least if we would know all of the parent neurons or all of the Markov blanket, technically speaking, then we could really make causal assumptions about these neurons. Okay, so I already established that this model here, this Hebb model, is a graph. And that means that we have these edges connecting the nodes. And when we have this combined effect, I can't do it with this standard graph language, so I have to invent this. And so in mathematical language, we call this a hyper edge. So it's not a normal edge, it's an edge that's connecting not 2 nodes, but 3 nodes. And so this is what we call a hyper edge. And the moment you have a hyper edge, we're not dealing with a graph anymore, but a hypergraph. And so all that means is that we're having n-area interactions here, in this case, ternary interactions. And so there's a lot of literature on this already. In fact, it's a very hot research topic. And so if we just stick to triadic or ternary interactions, people have pointed out that actually we're quite familiar with this. This is what we call modulation. So if you have, for example, neuron A innervating neuron C, and there's another neuron that is modulating them, you can think of that as some ternary interactions. There's also a lot of work on what is a hypergraph and how do we visualize it. So this right here is the exact same way as I showed it before. So this is what we call a rubber band. And so it's just showing these 3 things are interacting. So rather than this arrow right here, it's just still a hyperedge. It's not a directed hyperedge. It doesn't show you which way the causality goes, but it connects them in this ternary or triadic way. And we could indicate at least here the strength as the causal strength as the width of this rubber band. But again, we don't have directionality. So here's another way to do it. So this would, again, just be a different way of showing an hyperedge. This is taken from category theory. We would call this that upper red. These are just different ways to do it. And I'm just pointing out that this is a lot of work that exists already. And I'm also standing on the shoulders of giants in that using these kind of tools in causal analysis is not new. I'm going to point out mostly the paper here by Matteo Grasso from Giulio Tononi's lab that was published in Nature Neuroscience 2 years ago, where they pointed out that might be high time for us in systems neuroscience to take this causal root and to actually do that and look at higher order causal interactions. And they heavily lend from the mathematics that Giulio Tononi's lab has developed for integrated information theory. And so that's also what I will do today. So note that has nothing to do with consciousness. We're just taking these mathematical tools that are developed to look at causal interactions and see if we can do what we want to do. Lastly, I want to mention work from Angus Liang from now Toshiya's lab. And they were the first, to my knowledge, that published last year in PLOS Computational Biology for the first time that you can do that. You can take these mathematical tools, you can apply it to neuronal data. They did it to neuronal data in the fruit fly, and they found interesting, promising results. Okay, so first, I think I have to briefly convince you probably why we could be doing this, to collect data, look at neural data and causality. And so what I will ask is, first of all, we have to make some assumptions. We have to make some trades-offs. So I will keep things very simple. So let's just look at ternary examples where we have, let's say, 2 input neurons A and B and output neuron C. And we just, again, very simple, keep it binary in the sense that these are either active or inactive, on or off, 0 or 1. So you could think of that as spiking or no spiking. This also works for other data, e.g. fMRI, you name it. Of course, you would just have to binarize it, let's say above the median or below the median. And I think that's no problem to do this, even though you might say, well, there are neurons, let's say in C. elegans, that actually do analog computing. They do not use spikes. They don't do binary computing. But so this is maybe a bigger, more philosophical point that we have not yet found infinity in nature. So in fact, all of the computers, everything we do is fundamentally resting on discrete math. Discrete math is a little underappreciated in its power. And it's fine. It's great, actually, to use discrete math and non-infinities. And so what that lends us is combinatorics, and that's a little bit the crux. That's the only crux that I will admit to, so that when even we're dealing with such a simple system, combinatorially, it already gets quite complicated. So you can think of an A, B, and C all be inactive, not firing any action potential, so there would be 0, 0, 0. If only C fires an action potential, it's 0, 0, 1, and so on. And so you can already see that we get a long list of different system states that could happen. So then all we have to do is look at what are these possible system states using combinatorics. And then we have to do a little bit of statistics. And we look at how frequently does each state follow by the other state. So we establish all possible states, which for these 3 neurons, which of them could be active or inactive. So we have a long list. And then we see if these states occur, how often does any of these other states or the same state reoccur. So then we get a likelihood, basically, of each state being followed by another state. So this, of course, is 100% correlational. This is just standard statistics. And it's okay because we're not talking about causality yet. But what we have at this point is a matrix. And this matrix has transition probabilities. So we call it a transition probability matrix, or a TPM is what it's called. And this is a more general thing. This is not specific to IIT. This is used in all kinds of these Markovian dynamics. So the interesting thing that IIT introduces is that it never ever goes beyond that. So note what I did here right there is that I said we take the state of all three neurons, let's say 0, 0, 0, and then we look at if the system is 0, 0, 0, how frequently does then the system transition to, let's say, 0, 0, 1 or 0, 1, 1. And that is key, because what we never do in this analysis is look at what individual neurons do. So we take these three neurons as a whole, and we keep them as a whole. So we say this is 0, 0, 0 is a thing, it's a whole. And then we see when does 0, 0, 1 happen? And again, we're not worried about the individual neurons. We keep track of them. We make sure that neuron C is always in the last position, but we're not analyzing neuron by neuron. What we're actually analyzing here is what the system as a whole does. And so I think that is key why this kind of technique allows for these higher order interactions. And it's different from some of the other approaches that are currently around in topology and topological data analysis, manifold analysis, where people start with pairwise correlations and then try to infer higher order correlations. This is not what's happening here. Okay, now you might say, that's all fine, I understand, but that doesn't tell us anything about causality. And again, I'm probably going to go a little bit more fast here, that if you have questions, we can talk about it at the end. So the first thing is, if we want to know causality, we should know the connectivity of these neurons. And so in this case, of course, we can either assume the connectivity, or we might even know the connectivity. And everything that I will talk from here on out, we will know the connectivity. And in this case, we started with HEB, we know the connectivity. It was established. We know that A is innervating C, B is innervating C. So that is what I showed in the beginning. So we know the connectivity. And that is, I already did it to you, the big trick, how we get to causality. Because if we know the connectivity, then we can already rule out a lot of other possibilities. So we know that it can only be that A is causally impacting C and B is causally infecting C, and no other combination is possible. So if we have that knowledge, if we have that power, what we can do is basically create an artificial or electronic copy, if you will, of the system that is truthful to it in its causal structure. Again, this relies on the fact that there can't be any other connections in the actual system that I wouldn't know of. Otherwise, it would fall apart. But if I do know the connectivity, I have now established a mathematical model, formally speaking, a structural causal model, and that tells me what the possible causal effects are. And you can probably see where I'm going with that. If I know what all the possible causal effects are, and I'm observing certain causal effects, and I also can find out what the counterfactuals are. So now you can see how I can make this leap from observational data to strong causal inference. It is resting in the fact that I already know the connectivity right here. And again, this was found several decades ago by Julia Pearl, who is granted to be one of the first theoreticians who realized that science can find a way out of associative, correlative thinking by having this wrong assumption that is all that you can do, that unless you intervene, you cannot find out what causality is. And again, the trick put in very simple terms right here is that there is, this again taken from meta work of Julia Pearl, and I just want to put your view to the right-hand side. What he said is, because you can never really, as a scientist, observe causality. You always, as a scientist, can only see the effects of causality. You can very well model what these effects are with these structural causal models, or SCMs. And that's how you can get away from purely associational thinking, from trying to infer causality from correlational data. You can even go beyond the interventional level. If you intervene in the system, you still might sometimes be wrong, as you very well know, because even if you give a certain pharmacological substance, it's still too complex as that you could fundamentally, absolutely black or white, find out what the causal effects are. But if you know all counterfactuals, if you know everything that could have happened, everything that could have happened but didn't happen, now you have the highest level of making causal inference. And that is possible by intervening post hoc. So yes, you have to be interventional, but you can now do the interventions on your structural causal model. And that's shown right here again, where you have a causal graph. So this is a little bit more complicated graph where X and C interact, and then there's another variable Y that are interacting. And what you can do is what Julia Pearl found out, you can mutilate these graphs, or you can cut them. And you can basically, in your mathematics, pretend that a certain causal interaction doesn't exist anymore. And if you do that, now you can find out what the counterfactuals are. And you can see if a certain causal effect exists, yes or no. And that's exactly the mathematics that I will apply here as well. So let's go back to the simple case of this very simple structural model. And it is established by the connectivity that I just gave us ad hoc. I declared that this is fine. We can ignore the rest that happens in the brain. And then all we have to do is mutilate or eliminate these edges and see what happens. And so mathematically this is done with the do calculus or the do operation shown right here, where you say basically, I do separate, in this case, A from C, and then see what happens. And so if I have a complete description of the system, I can do this in a computer. I do not have to do it in the system itself, because it's an electronic copy of the system. And another term for that would be marginalizing. And again, I don't want to get too deep, but here is a typical transition probability matrix that we could establish from neuron A on B onto C. Again, see how the rows and the columns is fundamentally always looking at the system as a whole, and I get these certain transition probability matrices. And then, so here would be a much more simple example where we look at neuron A's and B's and their transition probabilities, because there's a certain distribution of probabilities. And let's say I marginalize, I cut, or I mutilate one of these causal interactions. You can see that basically that would bring some of these probabilities down. It would simplify it. And so if I recompute the system now that I mutilated, I find there's a different set of probabilities. And it's really interesting because, first of all, that tells me that there was a causal interaction, because if whatever I mutilated right here would have been ineffective, there should have been no change. So it's a way to analyze a system for causality. And it also allows us now to quantify the effect, because all we have to do is come up with some kind of 2D measure that allows us to compare the distributions on the left with the contributions on the right. And so again, that's another trick that I'm going to use. So all that we're going to do, though, is we're going to use some of this not just with regular graphs, but we can also do this with these hyperedges. So we can also look at these higher causal interactions. We're now not anymore stuck with these pairwise interactions. And then we can compute these distances between these resulting probability distributions. So very simple do calculus where we marginalize or noise certain connections or even hyperconnections, recalculate the transition probabilities, and then quantify the difference. Now, I'm simplifying things here. This is not what I'm actually doing, but I'm trying to gesture or tell you a little bit that you get an idea of what the underlying logic is. And I think this is very powerful, because before you might have thought, well, we have studied synchrony, Alex. What are you on about? We have looked at a lot of neurons that are active at the same time. And that's what I'm showing you right here. That's a simple correlation between A and B. But I'm also showing you that these neurons, they would give you the exact same result in a different structured causal model. So if neuron A and neuron B are connected with a gap junction and they're synchronously active, and only neuron B activates neuron C, you might find very similar results. But of course, it's a different causal model. And that's what we know the problem is with correlations, that there's many, many causal structures that give rise to the same correlations. And then one more, as I said, yes, okay, maybe we can do correlations with 2 neurons, but what about 3 neurons, 4 neurons, 5, and so on? What Pearson correlation coefficients do you now want to compute between these 3 neurons and go a higher level, right? So there's a problem here if we're just stuck to correlation. So the beauty is that we're not doing it, we're going higher level, and at the same time, we're leaving correlations behind and we're going to causality. And so now I'm going to show you a quick simulation with a simple computer model, exactly as you saw right here. I just wrote artificial neurons. You can see their activity right here. So they're Poisson distributed. You see neuron A, neuron B. And the causal structure of this model is such that if neuron A fires, a certain probability neuron C fires. If B fires, a certain probability C fires. But if A and B fire, the probability goes way up. And if I compute these spike trains into spike density functions, and then I compute cross correlograms, I just want to point out right here that you will find this very typical pattern that there's a peak that is preceding or following when one or the other neuron is active. And that's because the symmetrical relationship that you have in cross covariations is given by the formula. But that's indicative, and it's going to be interesting in a moment, that you have such a connectivity. Okay, then I'm doing what I told you. So I'm taking this it's fully randomized time series. Each time I run the code, it's differently. And I look at the states, right? So is all 3 neurons active, all of only one of them, which one? And so what I get is a new time series, but it's now not individual neurons. It's all 3 neurons, and you can see them in which state they are progressing. Now, you might still say, well, it's still observational data. And in the end, you're saying that you really know much about the system, but you need a lot of data for that. And that's true. But what I can do is I compute the transition probability matrix, and then I can add more and more data, and I can see, does it converge? And so that's what I'm showing you right here. So if I'm adding more and more data, in this case, this is picked from a slightly different analysis, but it should get the point across, that the transition probability matrix, after I have a certain bunch of observations, it asymptotes towards a certain transition probability matrix. So again, it's a little bit of an assumption, but I'm assuming if I have a certain number of observations, that the transition probability matrix that I have is a very good, if not complete, description of the system. This is just another way of looking at the transition probability matrix. And you can see for this individual system, if you're fully tuned in at this point, you will see immediately, yes, this system is a neural system. So most of the time, it will be 0, 0, 0. They're mostly off, these neurons. And that also means that the state that's following off is probably also that they're off. So the highest probability is they're off, and then they're followed off. This is what you see in the top leftmost corner. But you can also see that when neuron A and B, 1, 1, are active, or also when neuron A, B, and C are active, 1, 1, 1 at the bottom, doesn't matter because in both cases neuron A and B are active. You can see that now you have an increased probability that in the next step you have 0, 0, 1, which is that neuron C is active. So this does exactly what we thought it does. And now I'm going to run the analysis. So again, without going deeply into the analysis, I'm just going to show you the results. And I want to highlight that what I can do is I can look at, first of all, how many causators, and I would call this mechanisms, how many things are causally active at a time. And so that's counting first order, second order, and third order. And you can see on average that is 6 each time it's triggered, in this case, when neuron A and B are active. But more interestingly, what this algorithm pulls out is that the average order of mechanisms when neuron A and B are active at the same time is 3. It's showing you it's a ternary interaction. And when I compare that same data and I look at, let's say, neuron B and C are active, or neuron A and C are active, it correctly shows you that now the average goes down to 2, which is exactly what we would have expected. So this seems to work. Well, what about this example where now we have more neurons? So neuron A, B, and C, same thing. They're all active. I use the same logic, and I'm going to go very quickly. It works even better. So now you see that we get not ternary, but quaternary interactions, and you see that any of the other conditions doesn't have it. So very briefly, in the last couple of minutes, where am I going with this? So what I tried to convince you is that this is important. There's a technique, mathematical technique, that might be interesting for all of us, that might be interesting in these questions. And I want to really bring it back to neuroscience. So I decided to look into C. elegans for one reason only, because I made this point that I need to know all of the parents of a neuron to really know its causal structure. And that is the case in C. elegans. So C. elegans, as you know, is very small. It's a great model organism. These are all the neurons in a male C. elegans. There's about 1,033 of those. And all the connections are exactly known. They're shown right here. So the whole connectome is meticulously known. So I know exactly the causal structure of the whole system. There's all kinds of web tools that allow me to do it. And in fact, the latest papers, if you're wondering why are there all of a sudden all of these C. elegans papers in Nature in the last couple of months, is because they can do something that's really cool. So what you see in the top left corner is a C. elegans worm moving through agar. And so they can genetically modify that animal so that they can do 2-photon imaging while the worm is moving. And they can image hundreds of the CNS neurons at a time while the animal is moving. And you see this on the right-hand side. So you can collect neural data while the animal is active, not just of one, not of two, not of hundreds of neurons at the same time, and you can relate it to behavior. And so, you can feed that machine learning algorithms, and you can find out which neurons let us predict if the worm is going to move to the left or the right or back or forth. And all of these data are freely available on the web. And so all I'm going to do is I'm going to combine that. So this right here, for example, is all these neurons, they tend to have 4-letter acronyms. So AIBL is a very famous neuron that neuroscientists like in C. elegans, and you can see all of the parents and also the descendants of that neuron. And so I can take these neurons out of the database, and then I can look while the worm is moving, what are they doing? Now, there's some caveats here. As I said before, C. elegans neurons, most of them are non-spiky. And what I'm looking here is calcium imaging. But I told you that I can also binarize these data. So I did that. And so just pointing out here, with these neurons, I would have expected to find this correlational structure again with these peaks preceding. That's exactly what I find. So there's indication that these techniques, I'm not using any fancy technique here, just looking at the time series, cross-correlational, we get the result that we expected. We see that connectivity structure. Okay, I'm going to look at a different neuron right here. That is a CEPVR. And I'm going to take a triangle out of those. So CEPVL and CEPDR. And now, again, looking at correlational structure, just making sure that, yes, hopefully they are connected. You see that technically speaking, there's more going on right here. and run the fancy technique that I just talked about. And again, long story short, I get exactly the result that I was hoping for, that each time the input neurons are active at the same time, we get the indication that there's a ternary interaction. So this is now real data, not computational data. This is actual data, and it should take away a lot of the concerns that you might have had if I would have shown the IGN neurons in a primate or, let's say, a mouse. So I know the connectivity of these neurons is under my control, and I still find what I was looking for. So it looks Hebb was right. The brain is a network that's more complex than a binary graph. It matters when neurons active at the same time. That is going beyond our typical pairwise binary thinking. And that is important because I believe that might mean that we probably don't fully understand that brain is a complex dynamic system if we keep sticking to these pairwise mathematics. And so this is an invitation to everybody to higher arity mathematics and maybe join us in this effort, or also take these tools as far as they establish. As I said, all of the code is freely available, and think if this might be interesting for your data as well. So I would be very happy to collaborate or even just provide input for anybody who has interest in using this and sees value in that as well. That is pretty much it. And thank you again, everybody, for making it to the Zoom.